Okay, question 2 it is. Your question 2 was your microbiology question. So it's giving you a couple of images to get you started with. It's just literally looking at size, shape, staining. It tells you that gram staining has been used. So the first picture is more to be used with the shapes question and the gram staining itself. And it's even got the lovely labeling in case the color vision is a bit of an issue here. So first question here, state the names given to the shapes of bacteria. You need to have the official names. I can't have spherical and pill shaped. I cannot have those, I'm afraid. So I need the coccus, bacillus, and spirillum. With the spirillum, I have been a little bit lenient because spirale is kind of fine as well. So just use the official words that go with the labeling of those. Then the following one, state and explain what the results. Here we go. Again, you can see this here, state and explain 2 marks. State part. What are the results of the staining showing? What are the results staining showing? And image 2.2 indicates about the structure. Okay, so if it tells you about D and E, what does it state what it says, D and E? Basically, is D gram positive or is E gram positive? Which one's gram positive, which was gram negative? That's going to be your first memory, sort of trying to get to your memory aid. And if you remember, the gram positives are purple because they have plenty of peptidoglycan. Obviously, you can't use the plenty. You'd have to replace it with a thick layer of peptidoglycan, which then holds onto that crystal violet stain. That was the state, which one is which one, and the bacteria D are the ones that are purple, so the D are gram positive, the bacteria E are the gram negative ones. And then what does it tell you about the structure of the cell walls? So gram positive D will have a thick layer of peptidoglycan, however, no layers above it. And gram E will have a thin layer of peptidoglycan. However, that lipopolysaccharide layer goes over the top of it. I know that I said lipid layer when teaching a lot and said comparing it to an extra cell membrane, but you do need the official wording here with the lipopolysaccharide or lipoprotein layer as an additional layer above the thin peptidoglycan layer. Here in the mark scheme, you do not actually have to mention the thin peptidoglycan layer. And even for the positive, it lets you get away by stating that by not having to state there's an additional layer above that. This one here, and I've seen this so many times, but going back to your core concepts, where in biochemistry is this element needed? It gives it to you with sulfur, and you'd have to refer to lysine in proteins. It gives it to you for magnesium, and you'd have to refer to it as chlorophyll, center of chlorophyll. In this case, why would microorganisms need nitrogen? What biochemistry do you know that requires nitrogen? Proteins spring to mind. Any of the nucleic acids spring to mind. For the phosphorus, you'd need to go more along the lines of either the actual nucleic acids or the phospholipid bilayer to have those in there. And that literally that's your first part of biochemistry, which you did now nearly a year and a half ago. Last bit of the question, which kind of gives you that whole layout, the whole setup. So the nutrient agar can be modified. So that is expecting you to kind of have a little thought process and read and apply what you know about bacteria to that situation. It will tell you that one here is a salt agar. Whoever's it is, somebody has invented a salt containing agar, which contains a mannitol. You're thinking by now should kind of click in anything that's got ol at the end can potentially contain carbohydrates and is the source of nutrition for that for those bacteria, and a pH indicator. Okay, so you've got that already. So some color change will be identified from that. It tells you that some species can break down that mannitol, whatever it is, okay? If it's all at the end, it should know it's gold by now. Which those bacteria that can break it down will change it from pink to yellow. So if you know that there was a color change, you'll know that that particular bacteria could access the mannitol. You've got 3 different bacteria species named beautifully in italics. I'm surprised your mark scheme has not specifically written down that your answer has to be in italics. So you got away with that one, some of you. It gives you a lot of information here about the gram staining. Are they halophiles or not? Are they salt loving? So you would automatically have to figure out from this table that the first two, your Staph aureus and your Staph epidermis, they are halophile, they are salt loving, and they should potentially do well. It also tells you breakdown products of that mannitol which produce an acid. Okay, so Staph aureus is producing an acid. This will go together with pH indicator is going to do something. pH indicator is going to change pink to yellow. Okay, and last but not least, here you have got the actual agar plate where it is implying by the color that Z is the one that has changed. Y and X are the ones that are not changing color in the agar. So no response from a pH angle, but it also tells you that Y hasn't actually started growing. Which one hasn't started growing? The ones that cannot live on a salty environment haven't started growing. This whole thing is a combining the information from your table, from your text, and from the picture together so that you can then fill this table in. Okay. Oh, I didn't even go into the gram-positive and negative staining because that is another line of information here. So you've got the gram-positive and negative. You've literally got it here as a last bit of information to, once you've identified, add the gram-positive and gram-negative staining into this here. Over to the mark scheme. If you need a little while with the mark scheme here, just pause the video that you've got that in front of you.